In this video, we'll be talking about the beginning of protein trafficking in the endoplasmic reticulum, and how and why vesicles form from the endoplasmic reticulum. As we explained in the last video series, the proteins are translated in the endoplasmic reticulum. From there are two options for the protein. If a protein is hydrophobic and has a certain amino acid sequence, it travels to the membrane of the endoplasmic reticulum and becomes part of the ER membrane. However, if a protein has a different amino acid sequence, it will remain in the lumen of the ER. From there, vesicle formation occurs. A vesicle forms by budding off a piece of the ER. A part of the ER membrane becomes the vesicle membrane, and the respective piece of the lumen of the ER becomes the inside of the vesicle. Therefore, the proteins in the ER membrane become proteins in the vesicle membrane, and the proteins in the lumen of the ER are inside the vesicle. After a vesicle is formed, it moves to the cis side of the Golgi apparatus, which is the side closest to the ER. An important thing to note is that proteins cannot leave the ER unless they are properly folded and assembled. To ensure this, chaperone proteins, like BIP or calnexin, bind to misfolded or incompletely assembled proteins. The chaperone proteins prevent these improper proteins from leaving by covering up the exit signals or anchoring the proteins in the ER. These proteins are then transported back to the cytosol, where they can be degraded by proteasomes. This step is extremely important because incorrect proteins could interfere with normal proteins and damage the cell. Incorrect folding or assembling happens very often with proteins, so cells must produce copious amounts of proteins because many will be incorrect and therefore destroyed.